My name is Marina Douglas, and today I'm going to be talking about the topic of epigenetics. What is epigenetics? Epigenetics literally means above or on top of genes, but the actual meaning of it is it refers to the external modifications to DNA that turns genes on or off within our phenotype. Epigenetic modifications do not change the DNA sequence itself, but instead changes how cells read these genes, and this process of reading the genes causes it to be expressed within our phenotype, which is what I'll explain later on. In order to understand how the epigenetic processes work, we must first understand how DNA works. So DNA stands for deoxyribonucleic acid. It is in a double helix structure. It has 4 nucleotide base pairs, adenine, guanine, cytosine, and thymine. Our genotype is our DNA sequence or our genetic identity, and our phenotype is how we present our genes, such as our hair color and our eye color. DNA, when it is stored, it is wrapped around proteins called histones, and this process creates structures called nucleosomes. And nucleosomes are then organized to form another structure called chromatin, and a chromatin is the basic building block for our chromosomes. The way that DNA is wrapped around a histone essentially determines its accessibility to the tRNA, and the more accessible it is, the higher probability that it will be expressed in our phenotype, but the less accessible it is, it'll have a lower probability of being expressed in our phenotype. So the two main epigenetic processes that we know of are called DNA methylation and histone modifications. DNA methylation occurs when the cytosine-guanine nucleotide pairs within a DNA strand are silenced. That means that they are not expressed in our phenotype when a methyl group is added to them. However, this process is reversible, and by removing this methyl group from the cytosine-guanine pair, this gene is able to then again be expressed within our phenotype. The next process is called histone modifications. Histone proteins have exposed parts to them called tails, and these tails are highly vulnerable to chemical changes in their environment because these chemical changes essentially alter the chromatin structure, and this alteration determines which parts of the DNA sequence are accessible to the tRNA, and therefore which ones are going to be expressed in our phenotype and which ones aren't going to be expressed in our phenotype. So some factors that affect our epigenome are disease, diet, childhood experiences, parental attentiveness, exposure to chemicals, and exposure to drugs. As you can see, the epigenome tries to bring together the nature and nurture and how it affects us physiologically and psychologically. I'll talk about some of these effects later on in the PowerPoint. The clinical applications of epigenetics are medications for mental illness. Parnate is a medication that's used on individuals who have depression, and lithium is another medication which is used for individuals who have schizophrenia. These two medications affect a protein called demethylase, which inhibits gene methylation. So how this works is that it'll affect the demethylase so that the genes which code for depression and schizophrenia will be able to be turned off, and therefore will not be expressed in that person's phenotype, and therefore it'll either reverse or greatly improve upon that person's side effects from their depression or schizophrenia. So how epigenetics affects our psychology. So the two main areas of study of epigenetics and psychology are depression and schizophrenia. In a rat study, it showed that rats who had depression had a low concentration of something called BDNF, which stands for brain-derived neurotrophic factor, and it is responsible for growth, maintenance, and maturation of cells. So this low BDNF concentration was attributed to histone changes on the BDNF gene, which blocked its production. And so they used drug mediation, which affected the BDNF inhibitor and increased the production of BDNF and improved upon the effects of depression in these rats. The next is schizophrenia, and post-mortem specimens, such as humans and rats, showed a low concentration of a protein called renin, and this was caused by hypermethylation, so therefore the gene which codes for the production of renin was turned off because methyl groups were attached to it. And this is detrimental because this causes interruptions in pathways of the brain that involved GABA, which also affects our higher order functioning and cause these individuals to have schizophrenia when this happens. The effects of epigenetics. Epigenetics can cause psychological disorders, cancer, and physical disorders. There was a study done on rats that showed that mothers who had a disease which caused them to be overweight, have yellow fur, and be susceptible to diabetes, they basically changed their diet while they were pregnant with their fetuses, and this change in diet caused the fetuses to not have the gene which caused these issues. It can also cause abnormal phenotypes, and it can also affect future generations. Humans who experience depression or depressive states while they were pregnant, their children had a higher chance of developing depression than those who did not. The future of epigenetics. Resilience through adversity has been found in people that have epigenetic disorders. The epigenome is forever changing. Glucocorticoid receptors, these reverse and lessen the effects of trauma on the epigenome. And by studying epigenetics, we can prevent, early diagnose, and better treat psychological and behavioral disorders. Thank you for watching my presentation, and I hope you enjoyed it.